So I'm really delighted to welcome Dr. Rebecca Farley-Brown, or Beck as I know her, from the Field Studies Council, my former colleague. I invited Beck to talk today because of the fantastic guys that the FSC produces. And I, for one at least, was really excited to understand a bit more about what's going on behind the scenes, maybe what they've got coming up. So over to you, Beck. Looking forward to hearing your talk.
Okay, thanks, Kieran. Good morning, all. I'm Dr. Rebecca Farley-Brown. I've worked for the Field Studies Council now for nearly 25 years. During that time, I reckon I've developed well over 200 identification resources with partners and authors and various people. So, what is it and why do we do it? Well, what is it? That's often the first question we ask when we see something new. Now, sometimes what we see is bright and colourful and obvious. So it's got distinct patterns and colorations or shapes that allows us to simply use a guide and identify it or start to remember and learn what it is. Other times things are much more similar. Two nice black birds here. So, you've got less things to go on. Birders sometimes talk about jizz. That combination of features and characters that you build up the knowledge of and use to distinguish what makes that bird unique from another. Now, you can develop this from years of practice in the field. So, it may be years of experience, sort of you build up this knowledge. But these characters and features can also be used to develop identification guides. Hang on. So why is it important? Why do we need to put a name to things? Well, unless we know what it is, how do we know if it needs protecting? Take this little beetle here in the flower head. Last sighted in the UK in 1954. So if we come across one, how do we know what it is? Who out there has the knowledge of it and to say actually what it makes it unique? Now, traditionally, this was a role of the taxonomists from the museums and the specialist agencies. But over the last 40 years, there's been a huge decline in the number of those employed. So the actual knowledge base at that level has shrunk drastically. This has been recognised for quite a few years. This is NERC report from 1976. Back then, they'd already recognised the decline in taxonomic activity, the number of experts, the number of people doing work had reached the level where it's going to hamper ecological research and other and studies. So, what can you do? That's the things pointing out we've got impact at the very top level. Now, unfortunately, it's quite pressing. If you do a quick Google search, you'll find plenty of other similar reports have happened on probably about a 10-year cycle ever since. Comments are very similar, each one. We haven't got as many experts, we're not training people, where's the knowledge coming from? But actually, a total lack in the change of policies or funding support to actually reverse this. And back in 76, this led to the AIDGAP project at Field Studies Council. We took a different approach. Rather than looking at how to reverse this decline in taxonomy, we took the approach that if people have less skills, how are they going to identify things? So, as we know, training. One part of it was training. Run ID courses, have things that people can come to and develop their skills. And that led to some of the natural history courses over the years and now sort of online training as well. But the other big key area and what became the AIDGAP project was the production of the user-friendly keys. If people are identifying things, they've got less skills, they're less likely to be able to use the very technical guides that were available at the time. Now, AIDGAP's sort of quite well known just as that sort of branding acronym, but its sort of actual meaning is kind of got lost in time. Originally, we were concentrating on the difficult groups, those that there was a lack of key or no key, and that was hampering research. These days, our approach is to look at anything, there's something that needs an identification guide for, then we'll consider doing it. One of the things that was unique about the AIDGAP project at the time was actually testing things, going out, seeing if it works. Because the main thing for a guide is to ensure it's accurate. So there's main ways of doing this. Peer review, you send it out to other experts, see what they think, get their comments back. And that's a good way of checking to make sure it's accurate. But these experts aren't actually the people that are going to need to use the guide. So workshops, get a group of people together with the author, try some specimens, see how it keys out. And that's a useful way of doing it because you can check for accuracy. You know what specimens you've given them. You can see if they're actually getting through the key and getting these correct. The other is just to send out lots of copies to volunteers. Now, this is a great way of getting a lot of feedback back and testing it in various localities across the country. And you can get a lot of useful information. You don't actually know whether or not they ID'd the specimen correctly, but if you've got a lot of people all making comments about a similar part of the key or a similar couplet, then you know that's an area where people have got problems and you've got to go back and look at it. So originally, we used to just send out paper copies. This is an example from a crab test version from 1981. It's been out, someone's had a go at it in the field, they've written their comments all over it, and they've sent it back. These days, we're more like to send stuff out by just a PDF or share a file, and people email back comments or still sometimes send back an annotated version if that's what works for them. And the key bit of this is we can use those comments and so we can refine the guides and get them better before they actually go out and publish them. Hang on. You get a good guide, it can last. These are all AIDGAP guides that were produced over 30 years ago. And although there have been minor changes for sort of minor edits, they're updating taxonomy in places, a lot of them are very much as they were when they first came out. There's other guides that have been around for a long time. I recently got my hands on a copy of Norman Joy's A Practical Handbook of British Insects. It's over 80 years old, but it's still useful for certain things. Not all guides last. Sometimes, even though you've tested them, once it goes out for wider use, bits start coming up and you realise there are problems in the key. Other times they become outdated by changes in taxonomy, or simply other guides come out that supersede them. So, what makes a good guide? It's got to be accurate. Fundamental thing is we've got to be able to go out there, use it, and come back feeling pretty confident that we've identified something correctly and put the right name on it. And it's got to be the correct level for audience. Not every guide is suitable for every person. People are coming in from all backgrounds. We've got people using it for professional work and study. We've got those using it for leisure. Within both these, there's a huge range in skills and expertise. School children might need it for practical work for their exams. University students might be doing projects, but they might have very little experience. Then you get the amateur recorders through to the consultants and teaching staff and researchers who will generally have a much more in-depth knowledge, but also they will need to take identification properly to a much more detailed level, going down to species, whereas some of these groups might just be looking at family level. You've also got people coming in just doing it for fun. Now, some of these are the first light touch, those citizen science projects that are just getting people out and connecting with nature. So they're people who've got absolutely no background. But it can also be the real enthusiasts. And some of the people doing it for leisure are actually our national experts. We've noticed a real change in audience over the last 20 years. 20 years ago, or probably even a bit later now, this is the type of guide that university students and undergrads and sixth formers will be using for their study. Fairly traditional, fairly dry looking dichotomous keys, and they're taking things down to at least family level. So an example here is British and Water Vertebrates by Croft. Nowadays though, if you went out for a university group, they're more likely to be using freshwater name trail. Simple flowchart style, much more appealing and engaging to look at, and lets you to identify things quickly, but not to the same level. But in a way, this also reflects the change in actual fieldwork. Fieldwork's now more about going about collecting data, getting lots of samples done so you can go back and do a study, rather than just going out and looking at taxonomy, with the next size being of being able to name things. And sometimes things can be even simpler. This is an example from Opal, a citizen science project that ran about 15 years ago. And this was a simple pond health score. So people could go out, look at their pond, find a few key species, add up a score, and that will give them an idea of how healthy or not their pond was. And then will give them ideas of what they could do to improve it. So, as you can see there's a big change in presentation and the format we use. Even on advanced keys, so our AIDGAP level ones, there's a range in formats. Recent one to freshwater snails used a flowchart style, so the yes/no questions going through, but annotated and illustrated throughout. Another recent one, the AIDGAP grasses key, is a traditional die-cut format. We've got illustrations where we need them, but also we're putting further pictures on the other side and supporting information. Now, there's various ways of doing things, various looks, but at the end of the day, one of the key things you've also got to remember as well is the cost. There's no point producing a really good guide that's too expensive for people to buy. You've got to ensure that what you've produced at the end of the day is going to get used. Now, seeing there, we've got a lot of levels, a lot of things. These days, we don't just produce one guide to a topic, we might produce several. So if you come along, you may start on a citizen science project or an event or survey and start looking at things. You'll need a guide to put a name to it. So it might be a simple mini guide like these postcard size ones we do, or one of our range of WildIDs. But by that, you might look at it, a bit of things, maybe one of those studies in the garden or the quick bug hunts in the local park, and you've found a woodlice. Some people, that's it, you put a name on it. It's no longer just a bug or a creepy crawly, it's a woodlice. That might start an interest. So if you want to find out a bit more, actually, we've got a guide that will help people identify 12 common species. So they're starting to build up knowledge and experience. They can then move on to a more detailed guide, such as the AIDGAP one, which covers all 37 species found in the UK. Now, along the way to get there, they might need further help. They might go on courses, they might go on workshops, they might look for training events. And then, of course, as you get more knowledge, you might want more detailed guides. You might want to look at the atlas, you might want to look at a much more advanced guides. They're like the Linnean series of synopses. And hopefully, once we've got people at this top level, looking at the detail and the specialist, then we're also feeding them into recording and starting to get records of what people find and where they see them. So we can do some of this ID, but we can't do it on our own. The whole support to get people up this system requires us all to work together and work with partners. And that's one of the things that you see today. There's a range of us here all working on these topics. Oh, forgot to say. And about 95% of what we sell is this bottom end, the WildID, the gaining knowledge end, the starting out. About 5% is at the AIDGAP level, where people are starting to ID to species level and get knowledge at more detail. And about 1% is at that expert level. So we've got a wealth of people dipping their toes in or starting to get an interest. And the real challenge for all of us here is how we move people up from that first getting an interest and getting people started to develop their knowledge and get them involved in recording and other work. So if you're working on a key, what are some of the common problems? What are the pitfalls we will want to try and avoid? Well, ambiguous terms. Has it got more than 2 teeth or has it got less than 2 teeth? This is a classic example back from what started this AIDGAP back in 81. A quarter of the testers went wrong at this point. What we should have had is a statement of never than 2 teeth. People took more or less to mean about. The other problem is relative terms. Small. How small? Large. How large? Larger than species X. Comments like that are fine if you're working alongside a collection and you've got the species there to compare to, but if you're out in the field with just that one specimen you've found, that isn't going to be helpful. So, sizes, actual real sizes and ratios are really useful, but you've got to be careful here, because what do you do if there's overlap? Wing length 11 to 20? Well, if you're wing length at 19, it could fit into either of these options. Likewise, leaf hairy, but compared to leaf hairless or slightly hairy, well, there's a bit of vague overlap there. Where is the division between the two? So both of those are points where people would start going wrong in a key. Now, we're full of it in botany and entomology and other disciplines of jargon. We like lots of technical terms. These are just some examples of beetle colour descriptions that are used in various keys. And I borrowed this from a crib sheet I found of Roger Key. Now, if you don't know these terms, you can't access the keys because you're going through them and you don't know what it means. That's gone a bit wrong. Sometimes things can be quite easy. If you've got a colour separating a yellow flower from a white flower, good and straightforward. But colour can also be notoriously difficult. What you should have been seeing in the middle of the page here that's just gone wrong is a bunch of little fungi, toadstools, all in different shades of purple and red. Now, the 3 colours at the bottom here, pale pink to lilac or red, purplish, reddish clay colour, are all taken from a fungi key. Could you use those terms to separate out what you see in the field? There's a large sort of overlap there, but they're also very subjective. Purplish, that type of perception of how purplish is, or whether it's pinkish or lilac, we've all got a different idea and visualise colour in a different way. Might seem great. We've got a lovely bunch of snails here. Lovely range. Some very yellow ones. We could take those out. All very striped ones here. Maybe we can separate those out. But actually, they're all the same species. What we've got here is the brown-lipped snail. Huge amount of variation in colour form within it. Also very similar in colour and patterning to the white-lipped snail. So when you're working on these, you're actually looking at the shell features. How relative height of the spires, how many number of walls there are, and features like that. So you go down there and you're actually looking at the actual features of how the shell is built up and put together rather than the colours and patterning within it. Now, if you think snails were tricky, try slugs. Again, a huge amount of colour variation in each one. But unlike a snail, there's nothing firm and hard there that you can go and measure. So when we're working on the slug guide, that put up a whole other challenge. For this one, we used things like size. We looked at the tentacles and the tentacle colour. We looked at the patterning and variations. We even looked at features like the colour of the mucus they produce. And for those, we need a whole character table of features to help people separate out one from another. UK ladybirds. We're quite handy in the UK. Our ladybirds are fairly straightforward, the common ones to identify. The 7-spot, the 2-spot, the 22-spot ladybird. They've all got handy names that remind us of what we're looking for. But then we've got things like the harlequin. The harlequin has a huge range of colour forms. Now, the harlequin first arrived in the UK in 2004. It's an invasive species. They come over, it outcompetes our native ladybirds for food and will also predate on their eggs and larvae. At the time, it hit the press and got some quite good coverage. And we worked with some colleagues at what's now CHUK to help track the spread. They were sort of looking at trying to map it as it spread up through the UK. And we produced a little mini guide to help promote their survey. So it shows some of our common ladybirds, but also a couple of the common forms of the harlequin. Also working with the same group then, we produced a guide to ladybirds of the UK. And by now, we'd put on 4 common forms of the harlequin that they might find. And this was also, all this work was linked to some recording apps as well. And from that, they successfully monitored and tracked the spread. It's now widespread and common. It's here to stay. So choosing best characters. You want characters that are easy to describe, and you might want to make sure they're appropriate for the potential group. Now, sometimes we need technical terms. Sometimes we just can use fairly straightforward ones. Whatever you're doing, start off by making sure it's clear that what you mean. So an annotated drawing showing the key features is useful, or even just take a good photograph and label it up. We've got an example here from the Caddis key. From this, you're going to have to use the technical terms. People are going to need to know exactly what they're looking for. Illustrate. A good illustration can save 1,000 words. So rather than trying to describe something, just put an illustration there, but also put it in the right place. When the AIDGAP guides first came out, we started putting the images alongside the couplet. Now, due to the printing technology at the time, it was far more usual to find all the images were stuck in plates at the back of the book. I mean, this goes back to the paste up area. I've got examples back up in the archive, where people have literally cut and pasted pictures out, put them against typed up typewriter style text, and then it went off and was photographed and became part of a prints process. These days we've got desktop publishing. Putting images in place, moving them around, manipulating things is far easier, and we've got far more options of what we can do. One way I quite like now is ones like this example from the Caddis. We've got the diagram, the line art showing what it looks like. We've also got a photograph of a real specimen, so we can start to see what's that probably going to really look like and what we're hunting for on our specimen. Are characters sex specific? Right, common blue butterfly. It's fairly obvious, it's blue, nice little butterfly there, but there's the female. It's more of a dingy brown. So if you're identifying these, you're probably going to, by using colour, you're going to have to key them out more than once. You also might find keys relying on genitalia. Now, for a lot of insects, you can get quite good, it's very good for taking that down for things like some of the fly species. You look at the genitalia to take it down to the species level. But if you're doing that, you're probably going to have 2 keys, because you can have, or key it out twice for the males and the females. Or for some keys, if you go up to the high level ones for some of the groups, actually, they only key out the males. There's not enough identification features and not enough work done yet to be able to separate out females. Another thing we've got issues of time of year. What about winter? Say I'm looking at plant guides. In the winter, I'm not going to find flowers. I'm going to have to look at the leaves. What about trees? Come winter, a lot of the ones in the UK drop their leaves. So if you're looking at trees in the winter, you're going to have guides for it. So you want to look at features like buds and twigs and the bark. And we've got a couple of guides out in the AIDGAP range for that. We've got a winter trees guide and dichotomous winter trees guide, both of which are pitched at slightly different levels. One to get you started, one when you want to take into more detail. And then what about birds? How about my black-headed gull here? Well, it's missing its key black head feature, because this is the winter plumage. You've got breeding plumages, you've got juveniles, you've got molting forms. So sometimes you've got to either be very specific. This is a guide for birds in summer, or you're going to have to make sure you've got examples on your keying out or giving identification for all the types of forms people are going to see. Got an example here of the caddisfly guide back in 2007. Now, little caddisflies, tiny little things, turn up a lot in moth traps. So when we did this, we've got a lot of interest, and over 900 test copies went out. We normally think of about 100, so this really did take off. And they range from really inexperienced people who'd never seen a caddis, well, never looked at a caddis before, right through to the experts. We've got good feedback. It's good layout. We like the nice illustrations. But it also showed that there were flaws in the key. And one of the problems here was the character size. Now, one of the features you can look at for identification that's very reliable are the little spikes on the legs. And these are called spurs and spines. Different terms for ones in different parts of the leg. Now, you've got an insect here that's 5 to 20 mil long, so you can imagine the size of the leg and then the size of the spurs. So out in the field for hand lens, these were really difficult to see. So it got here issues. The characters weren't really appropriate for the audience of this guide. They were too small, they're difficult to see, and there was a lot of confusion of which is the spine and which is the spur. So we revised it, we refined it, and changed it instead, and the final key was featured at how you separated the families and used sort of a tabular format. We've got another guide to caddis. Just before COVID kicked off, we got involved in another key. The idea here was the challenge here, I suppose, was could you identify it from a photo? We're now all wandering around with smartphones, taking pictures of everything we see. So actually, could those be taken in instead of a voucher specimen? Could you look at the photo back when you got back home, and could actually you start to get reliable records for some species from photos? And this was a highly illustrated key because we were using images throughout, and supported by lots of illustrations of the species in their different forms. And for some species, yes, you could ID down. For others, you can't. So there's parts within this key where we say you can't go any further, and it'll refer you then to the more advanced type of key that you're going to need to use. Another thing where we have to think about audience with the Riverfly partnership work. Now, this was a little project working with anglers. They tend to be out on the river a lot. They've got their patch for their sport. They make flies that mimic what's actually appearing on the water, so the live flies, to catch fish. And there's always a lot of anecdotal evidence. There were more flies on the river when I was younger. There's not as many flies this year as there used to be. The thought was there, can they get these people interested? And also as well, could we get them to help monitor water quality? So they had a project going on, and they developed a 100-page manual to support it, and then a workshop where they took ID down to species level. But this was really a bit daunting. It was a bit too much for volunteers. They wanted to spend their time fishing. They're happy to give an hour a month, but that was about it. So we revised and worked with them, and we got a monitoring guide that was literally 4 panels each side. One side takes step by step you through the methodology so that it's always the same, it's always concise. The other side picked out key groups that they wanted them to monitor. So they had workshops, they had groups doing this, and they had links with the Environment Agency. So if you had species, they had trigger levels. If you found less than certain numbers of species, then you could have in contact with your your EA contact, and then they could look at it further. And actually, this actually led to some successful prosecutions. It flagged up whether it might be a potential issue. The agency could go and investigate more and find out what was happening. It also led to lots more projects, lots more people doing this. What started as a project in a couple of areas became a nationwide survey. There's been spin-offs like Urban Riverfly. There's been then a more advanced one that instead of doing the 8 groups, they took it to 33 groups. So there, one guide led to a lot more, but also all that work that had been put into that 100-page guide to start with, we distilled out, and we had things like the AIDGAP guide from mayflies, the AIDGAP guide from stoneflies came from it. Types of keys. It's not just dichotomous keys, you can see. There's lots of formats. We use flowcharts, we use tabular keys, but also these days there's online keys. We can use apps, we can even use AI. So you have to think about the format, what's the best way to do it, and what's the best way for your group. Little group of lichens here. This was an introductory key, just looking at some lichens on twigs. Picking up key characters, getting people to look at a small number of characters and use those to identify what's there. But also by looking at these small number of characters, it built up their skills so that when they started looking at other keys, they'd know the key features and be familiar with looking at them. So, online keys. There's examples out there. Lucid's been around for quite a long time, gone through various forms. All of these things, you go on, there's a list of characters, you start filling in what you've got. So an example here from a New Zealand grasses key, you start looking at it, what have I got? Leaf milk 7 mil, I've got a ligule, and as you put the features in, it starts to narrow down and show you a list of potential options. You have to kind of know which are the best features to put in. There's a huge list of options here for variation. It went way down the page. But when I started putting in gloom length, that's when it started to really narrow down and start to give me an idea of where I might be heading. Now, those are features of the flower, so tiny bits within the flower that I'm looking at. So sometimes with these type keys, it's a bit overwhelming. You're not going to put all the details in, and you have to have a bit of an idea of what are going to be the best ones for separating things out. Similar to the Lucid, IdentikIt was developed from another FSC project. Again, using characters to ID. On this one, as long as you can produce an Excel sheet with a full character data, so you need to put the character state in for all your species, then you can start to develop a key. And again, example here from Harvestman. As I put my body features in, it starts to give me an idea and list a probability of what's coming up. Now, what was quite useful with this one as well is you could start weighting it. So it'll start suggesting what's the next best character you use to help get your identification split. And then that helps you build up the knowledge of which are the features that are going to be best to look at. So there's various ones of these around. So and those are just two examples. There's a lot more options out there. So another example, unwinged beetles. We've got what? 4,000 is it beetles in the UK? I can't quite remember. Over 100 families of beetles in the UK. So if you're going to look at beetles, it's a bit daunting. How do you get started? A lot of the keys are for the specific families. But if you're new to it, how do you know which group to go to? So keys to split the families up are always really useful. So although Unwin wrote this key back in 1984, and it hasn't really been changed that much since, there's still a demand for it, and people still need that help to get to family level. So we looked at it and thought, it's time for an update. For all these things, we sort of start off optimistically. We know where a few issues are. We'll go in and fix those. Well, it soon became obvious that actually it needed a major overhaul. And this led to the thoughts of a new approach. Now, I passed this challenge to Stephen Docker, one of my colleagues, and he started putting together a character matrix, because that was his thinking of how he starts to work. And for all of us the same, when I was working on a little grasses guide, first thing I do is start to build up that Excel sheet, looking at the key characters to help me split things out. So he got a full big character database going, and he starts to look at it, and it starts, and looking at the keys and thinking, which features did people use to split this up? And it became obvious there's about 6 key features that we can use to get things down into major groups. Head, antennae, electrolength, things like that. And for each of these, there's a couple of various forms. So what the idea of this one is, you go in and for each of these, you pick a letter. So you look at it and think, which head form have I got? I've got plate A or B. You make a note of it. What do my antenna look like? Oh, I might be C or D here. And you work your way down. So antenna here, because lots of forms on each of these, you'd have illustrations showing you what you're looking for. And at the end of it, when you've looked at all those 6 key features, you get a code. So that lettered code then lets you go and find out what your options are. In this case here, it goes down to 2 families, but that still would take you to 100 species. So there you are, and from that, it helps split you down. Sometimes it might just take you to the 2 families, and you're going to separate it here and compare them. Other times, it might take you onto a further key. Now, because Steve has put together a character matrix, it meant he could also develop an online key. So just out now, it's fairly new. It's worth going out and looking at if you're interested in beetles and giving it a go, and please send us any feedback. This is the idea, a similar one. You go in, you put your body form in and those key features, and then it will start to narrow down what you've got, show you the possibilities, and then when you get to the certain ones, it'll give you a breakdown, more detail of the forms. No idea on time scale here, folks. So sometimes a guide hangs around for a long time, other times it moves and changes. Plant goals, these lovely little things we see appearing on plants. Sometimes they're pretty obvious, like this little pincushion goal here. Other times they might be a little more elusive as part of this leaf blade. Now, Plant Goal Society started back thinking in the 80s and started people off looking at the plant goals. And from that, to look at these various strange things, the little first draft keys appeared. Now, over time, people developed these further and started to produce guides. So I think it was then by 2002, we'd worked with Margaret Redfern and Peter Shirley developing what was then the first AIDGAP guide. Now, this went from sort of little leaflets and pamphlets they'd produced along the way to the first guide. Now, this was a chunky thing, 300 pages, 200 keys to these. It got people out. People started looking at them more. People started realising them in more places. So, 10 years on, we did an update because by then they found that there's a lot more. People are finding them on more plants. They're finding that some that they thought were one, but actually several different ones, and splits them down further species. So by now, we're up to 400 pages. I think we went from 200 to 300 keys. Again, keep the work going, keep the knowledge going. By 23, we again did another version. Now, 440 pages. We've added some new species. We've updated taxonomy. But what's interesting on this as well is then look at the recording. So we're taking the pincushion goal here, go and have a look at it by decade. So Plant Goal Society kicked off around the 80s. So this is where people were starting to think and look at things and make records. So as they're developing and testing the keys, more records are coming in. And then as we start producing and releasing the keys, suddenly a lot more people are looking at them, and we've got a lot more records coming in. So you see there that once you've got a good guide out, it can really help with recording. After all, if you can't name it, you can't record it. Columbular key produced back in 2007 by 2000, and at that point, these tiny little insects live in the soil. There was very little known about them because there wasn't a key to actually get them and identify them. So from 2007, when the key was finally published, don't forget its test version had been out before then. By 2011, we're starting to get coverage maps for it across the UK. So people are actually going out and studying it. 2 water beetles guides produced for the Royal Entomological Society, working with the authors on those, then led them producing 3 atlases showing distribution of beetles across the UK. So what's new? What's in development? Well, brand new grasshoppers is just at the printers now. We've got that out. So that covers the Orthoptera and Diphoptera from most of Britain. So from sort of a line across Oxford way upwards, it covers most of those. So it's just missing some of the sort of ones that are restricted to very warm spots down in the south. We've got weevils in development. Again, a very photograph-based key. Looking forward to seeing how that comes together. So we've got beetles in work. We'll have test versions of that bouncing around hopefully next year. We've got sedges out at the moment. The author who worked on the vegetative guide to grasses is now working on a similar one to vegetative sedges. And I've also got a wasps guide from the author of the recent leeches guide. So we've got a lot going on. It's always nice to have new things coming together. We've also got things at the WildID chart level. We've got another grasses fungi guide coming through this autumn as well. So if we're doing guides, think about practicalities. Is it needed? Sometimes you might look at it and say, actually, there's plenty of guides for that group. We don't need to work on that one. And if it's needed, who will use it? Key thing is who can write it, and that can often be a block. If you can't find the right author, or nobody's free at the moment, that might be that you've got to put it on hold and think about it for a bit longer. How to illustrate, line art, photographs, even coloured illustrations. It's a great variety of options there these days. Print or digital. It could go either way. If you are printing, where's it going to print? We do quite a work with overseas partners producing guides for overseas. So if it's going to be printed overseas, you've got to check that it's suitable for printing on their local printers. Also, think about the numbers you want. If you only want a few, then it's going to be a few hundred or even say 50. You're going to be using a digital printer. So that's going to be a smaller size press than say for our WildID fold out chart guides, which go to a LIFO press. We can't produce those WildID charts really for anything less than 1,000 print run. And then if you're making a guide, how are you going to distribute or sell it? So that's why people, you work with people, a lot of people come to us because we then can sell them and promote them through our website. Or if you're producing something for a project, make sure you know how you're going to get it to the people that are going to use it. And the key thing always is, what's the budget? Can you afford to produce it? Or are you going to have to maybe save money here? What are the other options to keep it within budget? And just to wrap up, a quick reminder, if we can't identify it, we can't protect it. We've got two similar birds here. Now, this is an example from New Zealand, but it made the UK Guardian back in 2015. There was a bit of a problem here, a bit of an issue happened. They were having a conservation project to try and promote one of them. There's only about 300 of the taki in the wild. It was critically endangered. Most of it on an island population. Problem was, there was the other puki, the bird there, that looked similar and used a similar habitat, and it was outcompeting this critically endangered one. So they decided on a kill. They were going to kill some of these birds to try and promote the other bird. Unfortunately, when it came to actually the kill, the wrong birds got shot because the hunters couldn't identify the difference between them. So we don't know and can't explain to people what makes one separate from the other. We can't really protect it. On this case, although they're similar, the nationally critically endangered one is a flightless bird. So on this case, just telling people the hunters only to shoot birds that were in flight could have saved this problem. Okay, that's me. Thank you for listening.